Hi everyone, I'm Marina Watowich. I'm a fourth year graduate student in the Snyder Macular Lab. I'm excited to share some of my recent work with you. Aging is the gradual degradation of many of the body's physiological processes and is associated with widespread inflammation and increased susceptibility to disease. This implicates the immune system as a key part of the breakdown in function that occurs with aging. The aging process is by no means static. Rates of aging and the onset of age-related phenotypes can vary between individuals in the population. For example, individuals who experience severe stress, like extreme natural disasters, are at higher risk for developing cardiovascular disease, which is the leading cause of death around the world. It's clear that the environment can contribute to variation in morbidity across a population, but how does a natural disaster actually get under the skin to affect age-related phenotypes, and can it actually accelerate the aging process? I investigated these questions in a population of free-ranging rhesus macaques that live on the island of Cayo Santiago in Puerto Rico. Cayo Santiago is an incredible study system. The field site's been around for over 80 years, and annual biological samples have been collected here for many years. Macaques are an excellent model organism in which to do translational aging research because they experience similar age-related declines in health as humans do, like frailty, cardiovascular disease, and inflammation. Cayo Santiago was devastated by Hurricane Maria in 2017, so much so that most of the vegetation you see in the top photo is still destroyed. Now, it's also important to note that there are no predators on the island, and while the macaques are free-ranging, they are provisioned daily. So our research team had collected whole blood in the 3 years before Hurricane Maria and 1 year after. And in total, we had 543 blood samples that I extracted RNA from and sequenced. 7,009 genes were detectably expressed, and I modeled differential gene expression using a mixed modeling framework and controlling for relatedness between individuals. About 1,100 genes were significantly associated with aging, 260 genes were significantly associated with experiencing Hurricane Maria. And this alone is exciting because not many studies have looked at and much less found a signature of natural disaster on gene expression. Now, even more than this, 104 genes were significantly associated with both aging and Hurricane Maria, which is over twice the number we would expect by chance. And because aging and Hurricane Maria affect many of the same genes, we next asked whether these factors affect expression of these genes in similar ways. This plot shows all 7,000 genes. Every gene is a point. The x-axis is the standardized effect sizes of the hurricane, so positive values mean that gene expression was higher after Hurricane Maria. The y-axis is the standardized effect size of age, and positive values indicate an increase in expression in older individuals. The positive association here is significant, meaning that exposure to the hurricane and older age alter the expression of many genes in similar ways. The genes highlighted in purple are the 104 age and hurricane-associated genes. 85% of significant genes are in quadrant 1 and 3, meaning that aging and the hurricane broadly similarly affect gene expression of all detected genes and those that are statistically significant. Genes in quadrant 1 are generally associated with inflammatory pathways, and genes in quadrant 3 are generally associated with protein folding pathways. Yet we still want to know if suffering in a natural disaster actually accelerates the aging process, and we can think about aging in two ways. Chronological age is the amount of time an individual has been alive. So take a middle-aged macaque, for example. Biological age is an estimation of how old the individual is based on, in this case, their gene expression. If an individual's gene expression is typical for their chronological age, their biological and chronological ages will match. If an individual's gene expression was more similar to that of chronologically older individuals, their biological age would be older than their chronological age, and this is what we would expect for hurricane-affected animals. Animals can also be biologically young. So to measure this, we used a transcriptional aging clock across adults that was developed in human peripheral blood samples, and individuals after the hurricane had gene expression profiles that were on average 2 years older than samples taken before the hurricane, which translates to 7 to 8 years in a human lifespan. Now, there's also a lot of variation in individuals' biological aging after the hurricane, and I'm beginning to investigate drivers of this variation and dig deeper into why gene expression might be changing in the way that we see. So I've recently begun looking at DNA methylation data generated using reduced representation by sulfite sequencing, and a similar pattern's emerging where more sites than expected are differentially methylated with both aging and Hurricane Maria. Now beyond this, aging and the hurricane also alter DNA methylation in similar ways, meaning that sites that have less methylation in older individuals tend to have less methylation in animals after the hurricane. These shared effects actually get stronger as we apply a more conservative false discovery rate threshold. Now we still want to understand the mechanisms by which Hurricane Maria may be affecting gene expression and downstream function. When we look at CpG sites that are differentially methylated with the hurricane, they're enriched in areas of the genome known to regulate gene expression. Other studies have shown long-lasting DNA methylation changes due to environmental insults, and DNA methylation in regions known to affect gene expression may indicate that Hurricane Maria could induce persistent effects on gene expression. So if DNA methylation did affect gene expression, then we would generally expect that reduced methylation is correlated with higher gene expression. And when we look at CpG sites and enhancers, this is exactly what we see. So here the x-axis is the standardized effect size of experiencing Hurricane Maria from our gene expression data. The y-axis is a standardized effect size of Hurricane Maria for our DNA methylation dataset. Now this might indicate that reduced DNA methylation is associated with increased expression. Conversely, that greater methylation is associated with reduced gene expression, exactly what we would expect to see. Yet, in order to get at causal effects, I next plan to test whether differential methylation mediates gene expression changes, and this will help disentangle the extent to which gene expression changes after Hurricane Maria are a result of differential gene expression within the cell or driven by DNA methylation changes. Additionally, we also know that social rank and integration can affect health in many social primate species, so I plan to investigate how different components of the social environment affect gene expression and DNA methylation. I'd like to thank my advisor, Noah Snyder-Mackler, our entire lab, our collaborators, the University of Washington and Arizona State University, the University of Puerto Rico, and the staff at the Caribbean Primate Research Center for working in very extreme conditions following Hurricane Maria. Thank you for watching.